as you heard, this will be like a ping-pong presentation, and we want to try together to convince you that ADA2 has probably, let's say, an additional function that is a lysosomal nuclease involved in the, let me just check in the nucleic acid sensing of myeloid cells. So, as many of you have heard this morning, ADA2 can manifest with hypogammaglobulinemia, and this hypogammaglobulinemia is responsive to treatment of inflammation. And this is the reason why we wanted to understand the mechanism, let's say, leading to this kind of physiopathology in ADA2. And then we started to analyze secondary lymphoid organs that are organs important to generate immunoglobulin together with the bone marrow. And then we started to analyze secondary lymphoid organ of a patient with ADA2 deficiency, and we observed a very strong infiltration of macrophages, indicating that macrophages are important in mediating tissue damage in this disease. And as you can see here, using different technologies, we observed that ADA2 is almost exclusively expressed in macrophages and monocytes. Here you see secondary lymphoid organs, so germinal centers with germinal center macrophages expressing ADA2. This is an immunofluorescence of peripheral blood and bone marrow, showing that only CD14 cells express monocytes. And this is an analysis of proteomic analysis, proteomic datasets in different immune cell subtypes, showing that only, let's say, in particular monocytes with difference in monocytes differentiation states. Monocytes, they are the cells that they express the highest level of ADA2. That's the reason why, let's say, to understand the physiopathological mechanism of lack of ADA2 into these cells, we sorted the cells from ADA2 deficient patients and from healthy control. And here, and then we perform a full spectrum proteomic analysis of CD14, forward side scatter selected monocytes. And as you can see here, we observed that the level of expression in the patients is constantly globally reduced, strongly reduced. It's interesting also to note that according to what we have discussed this morning, that there are two different kind of patients that do not express at all the protein, so the level of the protein is 0, and some that they do express the protein. And as confirmed for what has been already, let's say, just mentioned, we also observed in an unbiased way, so only monocytes, that these cells, they express a very strong and quite impressive increase in type 1 interferon response and related pathway like response to viruses. This is another kind of analysis. Here you can see this is the network analysis, so you can see the interferon response and the other responses, the other pathway related to interferon, and here you can see some of the protein that are more upregulated, and there are many of them into the pathway of interferon. And let's say in agreement with this very strong increase in the interferon signaling, we try to infect the cells with GFP coding viruses and monocytes always, so sorted monocytes from ADA2 deficients and wild-type patients, and we observe that the ADA2 deficient monocytes, they are strongly resistant to infection with a virus. Here this is a flow cytometric analysis of GFP infection 24 hours after exposure to virus. And, related to what we let's say discussed, what we have just heard, we also analyzed in the same data sets the expression of many other protein that are known to be causing in different interferonopathies like STREX1 and many other, and we did not observe any differences in the expression of significant differences in the expression of other proteins involved in causing type 1 interferonopathies. And interesting, let's say, clustering the analysis of some of the protein that were upregulated in ADA2 deficient patients, so patients that they do not express at all the protein, or patients that they do express a bit the protein, we observed a very nice correlation between what we call the ADA2 dim, so reduced the level of expression, or ADA2 null patients. So every time that you do not have the protein at all, you have a very higher and significantly higher expression of interferon signaling. Going back to the secondary lymphoid organ, so I told you that the protein is expressed in particular in macrophages. These are germinal center macrophages, and these are so-called tingible muddy macrophages that, as you know, they are cells that are actively phagocyzing. And as you can see here, the pathway of ADA2 in blue resembles phagolysosomal phagolysosomal vesicle. Therefore, we hypothesized that the protein could localize into the protein, into the lysosome, and to test this, we performed on the same tissue co-immunostaining with ADA2 and LAMP1, and we clearly have detected that the protein is actually contained into lysosomal vesicle, into secondary lymphoid organ. After this, I give the word to, to my colleague.
Okay, thank you. So I'm the biochemistry person here. And luckily we had another speaker here about talking about the glycans, so ADA2. And as you see on the left here, I don't know if this works, but anyway, the enzyme, the glycans are mannose-6 phosphorylated, and it means that it contains a sufficient structure to be recognized by the phosphotransferase in the cis-Golgi apparatus, so it becomes mannose-6 phosphorylated, and that's actually the sorting signal for transport to lysosomes. So it fits very well with a result by Michele on the macrophages, and it's located in the phagolysosomal compartment. And we get the same result, just here is a recombinant human enzyme, and here is the enzyme from porcine brain. And you see in both of them they are phosphorylated. You see also some glycans which are high in mannose but not phosphorylated, and it's probably because you have a dephosphorylation occurring in the lysosomes. So the degree of phosphorylation is less than 100%, is maybe around 30, 40, 50%. So the rest of the glycans are complex type glycans, and it might indicate that the enzyme is both transported to the lysosomes and secreted. If you look at the next slide, we have analyzed the glycans linked to all the glycosylation sites in ADA2. And in human, as you heard before, you have a glycosylation site N185, which is not evolutionarily conserved. And from the left to right, it is increasing processing of the glycans. And this glycan here, at least at N185, is not processed. It's a high mannose type glycan. It is reminiscent of its location in the cleft close to the PRB, which is a putative receptor binding domain of ADA2. So it's not mannose-6 phosphorylated. The three other glycosylation sites, which are evolutionarily conserved in vertebrates, they are all mannose-6 phosphorylated, although to a different degree. This site here, 174, is maybe 20% mannose-6 phosphorylated, while the two other sites here, 127 and 378, is mannose-6 phosphorylated. And very important here, you see the same type of glycan structures, both in the human enzyme and also in the porcine enzyme, so it's an evolutionary conserved post-translational modification. So we get to the question here about the glycans which are not mannose-6 phosphorylated, and do we have some secretion of the protein? So I compare the glycan structures that you find in a secretory protein. Here's a hyaluronic acid protein, which is contain unprocessed complex type glycans, which is very typical for secretory proteins. And here is so-called trimmed glycans, is paucimonocytic glycans, which are core-fucosylated and which you otherwise find only in insects, and they are trimmed in the lysosomes. If you compare lysosome alpha mannose, which is a lysosomal protein, with ADA2, you find the same profile. And you see also the same also with the mannose-6 phosphorylated glycans, that you have the same relation between these mannose-6 phosphorylated structures, both in aryl sulfatase B and also in ADA2. So it shows that when I purify the protein from porcine brain, all of the glycans, ADA2 that I purify, originate from the lysosomes. Of course, if the protein is lysosomal, it must have kind of function, which is related to the low pH of the lysosomes. And then here we have made a model based on the excellent structure by Savialo from 2010. And we see here that the PRB domain, putative receptor binding domain, which is absent in ADA1, actually has signature sequences by in silico measurements binding to DNA. And also you can look also at the catalytic site here. A big difference between ADA1 and ADA2 is that ADA1 is binding only to the purine unit, but ADA2 is binding both to the purine unit and also to the ribose unit. And then you're getting quite close to the phosphodiester linkage, and you have some kind of a possibility that actually the enzyme can cleave DNA. And as you see here, it's only one site here, so it must be a nicking endonuclease if it's a DNase. If you look at the next slide, and I will turn to the right side here, you can see that the enzyme is actually a nicking endonuclease at the low concentration of ADA2. This is a plasmid. It's a supercoiled DNA here. It's a nicked form here. And if you treat it with ADA2, the supercoiled form is going over to the nicked form, so it's a nicking endonuclease. If you increase the concentration of ADA2 or increase the incubation time, you get a complete degradation of the DNA. We have been doing a lot of studies to see that it is really the DNase activity that we find. And in this slide here, we have 3 STS pages, and they have exactly the same enzyme activity. And we can see that the bands here are this band here, this band up here. They are both related to the ADA activity. And if you do MS analysis, you see a very high score on ADA2. But the proteins with lower score, we can't see any protein that might, in theory, be cleaving DNA. So we are quite sure about this. And we also tried out on the recombinant enzyme, and it is also cleaving the DNA. So we think that this enzyme is a nicking endonuclease, and you can see also that the endonuclease activity has an acid pH optimum, which conforms to the enzyme being a lysosomal protein and functioning as an endonuclease. I think just now I take over to you. I hope it's fine.
Okay, so Ole, what in the previous slides, Ole showed you data about particular porcine brain ADA2. We wanted to confirm this data and further characterize the activity in human recombinant ADA2. Here is just to show you that ADA2 is actually upregulated by a treatment with type 1 interferon alpha, in particular mRNA expression, and also protein level. And this, I think that is also important when you take in mind when we do actually analyze the protein or the enzymatic activity in the functional tests, because according to what has been also mentioned before, according to how is the activity of the cells or the activation state of the cells, or which kind of cells you have in the peripheral blood, also what you find analyzing ADA2 can be different, because the expression of the protein is strongly up or down regulated according to different stimuli. Here you see interferon alpha, but we have different kind of data also with other toll-like receptor agonists. What you see here is actually a comparison with of the DNase activity of ADA1 and ADA2. That's why, because as Ole told you, we have seen, and we have, the model that Kristian Smulski, a colleague of us, performed, showed that double-strand DNA is binding to the PRB domain that is not present in ADA1. Therefore, we wanted actually to analyze if and to compare the activity of ADA1 and versus ADA2 in terms of DNA activity. This is threat analysis of DNA degradation, and as you can see, ADA1 has no activity at all, and ADA2 actually is confirmed to have a strong DNase activity at lysosomal pH. Another interesting observation is that how it happens for basically all the intracellular DNAs, the activity of the protein is increasing with the reduction of the salt concentration. And this is just to, let's say, this is just a message related again to what we discussed this morning, that the protein is, for example, one of the most important, the most frequent mutation in observed in the patient, that is the G47A, is lower expressed, but it maintains the protein, it maintains the DNase activity, and this can somehow explain the dose response. Moving to this, we just then went to some signaling analysis, and here I'm happy that the STING, let's say, pathway has been already introduced because I don't have to repeat it. But in this experiment, we observed that we directly measured the interferon alpha and beta secretion in ADA2 wild type and knockout cells, and we did not observe any differences when you stimulate with different Toll-like receptor agonists, but actually we did observe a strong increase in the interferon production when we stimulate the cells with double-strand DNA. And this strong increase of the interferon production after double-strand DNA exposure, it is reduced, so it is abolished when you overexpress ADA2, wild type ADA2, or when you double knockout together with ADA2, you double knockout also STING. Because as the colleague before told you that the STING activation is dependent by the pathway of CGAMP production, we also measured direct production of CGAMP in cells that are knockout for ADA2 or overexpressing ADA2, so ADA2 plus ADA2 knockout plus re-expression of ADA2. And as you can see here, when you do not have ADA2, you have a strong increase in the production of CGAMP that can be completely restored by overexpression of ADA2. And with this, I go to the conclusion that here we provide a multiple line of evidences that CCR1 encodes a protein, ADA2 I should have written actually, encodes a protein that acts as a rheostat for nucleic acid sensing of myeloid cells, and somehow they help to reconcile ADA2 function with the ADA2 immunopathologies, in particular with the interferon signal. And with this, it's all. And thank you.